What's up? We are back. It is Tuesday, April 7th, and we have made it to the end of the road with the neuron. We are going to be looking at neurotransmitters today. And I'm going to break this into parts because we have a bunch of different ones that we're going to look at and dive deep into. So the first thing that we have to do is we have to define this term neurotransmitter. Right? And you should be able to break it up, right? You have neuro, and then you have transmit, right? So it's a chemical substance, and it's going to be stimulating a neuron, whether it's going to be making it stimulate a neuron to move an effector or create another action potential. It's a chemical substance. It's released by a neuron to stimulate whatever it is. Effector, neuron, whatever's at the end of that axon terminal. So we're going to write a chemical substance released by the presynaptic neuron into the synapse. Now, that neurotransmitter gets into the synapse. Then what? A bunch of it is going to go onto receptors on a neuron or an effector, and it's going to make it do whatever job it's supposed to do. Move a muscle, cause an action potential, whatever it is. What about the rest of the stuff that's in there? It's got to go somewhere. So there's a few different things that happen. We're going to talk about two of them. What could happen to the neurotransmitter? So it could be reabsorbed by the neuron, the presynaptic neuron. And serotonin is one of the neurotransmitters that is reabsorbed. And we're going to talk about something called SSRIs, which is selective serotonin reuptake inhibitors. And we'll get into that when we get to serotonin. Also, we can have enzymes that break the neurotransmitter down. And you can see on my screen, I have acetylcholine, break down neurotransmitter. There's an enzyme called acetylcholine esterase that breaks down acetylcholine. That does not get reabsorbed. All right, so part one here, neurotransmitters. They're a chemical substance released by the neuron into the synapse. And then what happens to this neurotransmitter after it's released? It stimulates a neuron, stimulates an effector, gets reabsorbed by the presynaptic neuron, or it can get broken down by an enzyme. All right, let's get into acetylcholine. Be right back. All right, guys, so now we are going to get into the different neurotransmitters. So the first one we're going to do is the most common one, and that's going to be acetylcholine. Now, it's a long word to write, so we abbreviate that with ACH, acetylcholine. And so with acetylcholine, this is going to cause an action potential. It's going to cause those sodium channels to open. Sodium goes in, changes the membrane potential, and then you have your stimulus. There are different types of acetylcholine receptors. So let me write receptors here. Receptors, and they have a name. They're called cholinergic receptors. And there's two types. So we have ones called nicotinic. Does that sound familiar? Nicotinic. And then we have these ones called muscarinic. I'm not talking about muscarinic. It's like slow response, very specific. We don't need to worry about that. But we do, or we'll talk about nicotinic, but briefly. So, like a nicotinic. There you go, receptors. Sounds like nicotine, right? And it's true. Nicotine will go nicely into those receptors. It's kind of amazing if you think about that. And we could do a whole big project on it and why addiction happens when you're smoking cigarettes. And it's because of these receptors that are found all over. And acetylcholine being one of the most common neurotransmitter, that means a lot of your neurons have these receptors. So you're very receptive to nicotine. So don't smoke. Anyhow, these nicotinic receptors, right? One thing we need to know is that all somatic neurons release acetylcholine onto muscle cells, which will cause the muscle cells to contract. And let me get specific here, skeletal muscles. So these somatic neurons, they're going to, and those are the neurons that you control consciously, they're going to dump acetylcholine into muscle cells to make them do their thing. So if you remember, we had our motor end plate, the neuron that was going to attach to it. They're going to be dumping out acetylcholine, which is going to cause the calcium to come out of the sarcoplasmic reticulum. It's going to cause the troponin to be released from the tropomyosin and allow the myosin to attach to the actin and have that power stroke so that you can have motion. All right, so we need to know one more thing about acetylcholine. The enzyme that breaks it down is called acetylcholine esterase. And that's one word. I'm putting it as two. Esterase. And I think I spelled that right. Acetylcholine esterase. That ASE denotes that we have an enzyme. And so that's like a little Pac-Man if I was to draw that, or if I drew you a little neuron over here. Here's the axon terminal. And I have acetylcholine over here. We'll use some different colors. Here's my Pac-Man, or my acetylcholine esterase. It's going to go in there, eat up all that acetylcholine so it gets out of the synapse, so then you can have another action potential take place. All right, our next one is going to be serotonin. This is one of my favorites because it has to do with your mood. Be right back. Now we're going to be talking about serotonin. And I decided to change my color just because I got all red on this thing. I'll do a little blue now. So serotonin is an interesting one because it controls a lot of different stuff, right? So serotonin is a neurotransmitter. It is going to be regulating the following. One is going to be our mood, right? So you're happy, you're sad. That's the amount of serotonin that's hanging out in the synapse and being picked up by that postsynaptic neuron. Another piece is your appetite. It controls your hunger. Another one is going to be your memory, as well as learning. All of these are involved, serotonin is involved in. Another one here is sleep. So this is a really important neurotransmitter. And actually, having too little of this neurotransmitter causes problems with mood. And that should be obvious, right? And also results in many people carrying out risky behaviors. What is that? I don't know. For me, that'd be like going on a really big roller coaster. But other people can do crazy things. So it's also associated with risky behaviors. So I wanted to take a minute. I wanted to look at serotonin because it is important. That's what it looks like over there for you chemistry peeps out there. But we have these things called SSRIs. So it's something like Prozac, Zymbalta. Those are going to be antidepressants. And the SSRI is a selective serotonin reuptake inhibitor. So we're going to write that word down and we're going to look at it. And I'm going to pull this over here. And I'm going to write SSRI. So it's selective serotonin reuptake inhibitor. I'm writing inhibitor. It's like Liberty Bibberty. Inhibitor. So let's look at it. It's selective in the serotonin, right? It will not allow, it's going to inhibit the reuptake of that. So you have these things. If I draw you, I don't know what color we should use. We'll go with purple. So we draw you a Actually, I'm going to fix that a little bit. Here we go. We're going to draw a neuron over here, and then I'm going to use, let's use orange. Here we go. And then we're going to go back to purple. There we go. Okay. So these are called serotonin reuptake pumps. So what do they do? When the neuron releases that serotonin, the stuff that's left in that synapse that doesn't get taken up by that postsynaptic neuron, it's going to get pumped back into the neuron. So then it can be reused and reused and reused. The body's pretty insane like that. So what does an SSRI do? That SSRI, after you have the serotonin, what color should we make serotonin? What do you like? We'll go with, so limited on my colors. We'll go with this purplish magenta sort of color. So here is all of our serotonin over here. It's been released. You feel good. You're happy, but you want to stay that way. So what happens here is these serotonin reuptake inhibitors, they block it, and they keep that serotonin in the synapse. So then you can feel happier or whatever it is that it's doing for you. Are there addictive qualities to these things? There could be, right? Also, some of the problems that can occur is that since you have these things blocking and you have plenty of serotonin hanging out in the synapse, then the neuron doesn't make as much. And then sometimes you have to take another antidepressant along with your antidepressant to kind of boost or keep that serotonin in there. All right, so that's a little bit of serotonin. There's another little interesting tidbit. Serotonin reduces pain in the spinal cord. So if you have some sort of trauma, serotonin is released and it reduces the pain so you can get out of trouble or whatever. All right, we have, you know what, we could just jump into dopamine real quick. Dopamine, that is your brain's reward center. So that is what makes you feel happy. If you do something, your brain will reward you with dopamine. And then we are going to look at drugs next. I have this little picture here because when I was a kid, and I'm saying when I was a kid, there used to be a commercial and they'd have, this is your brain. And they would crack an egg, and they'd put it in the frying pan, and they'd say, this is your brain on drugs. All right, so we're going to come back to drugs and see how drugs affect our brain. All right, here's the last section of this. We're going to be looking at drugs, and we have two terms that we need to know, especially for Wednesday and Thursday. Wednesday and I think Wednesday and Thursday. Definitely Wednesday. We have drugs that could be agonists, and then we have drugs that could be antagonists. So with agonists, this is going to increase the effect of the neurotransmitter. Right, so that's going to make that effect more. Antagonists, that is going to do the exact opposite. That is gonna decrease the effect of the neurotransmitter. All right, so we have two ways to do that on both sides, and then we are done. So let's look at the first way. How can we do that? Well, we can increase the amount of calcium that goes into the synaptic bulb or the axon terminal to release more neurotransmitter, right? So if I just drew you that little picture over here, here's your calcium channel. If we increase the amount of calcium that's going in, we have our synaptic bulb that has our vesicle in it. We can cause more exocytosis. We can increase the amount of neurotransmitter in there. So we're going to increase the amount of calcium release to increase exocytosis of the vesicle in the axon terminal, which makes sense, right? The more calcium that you have going in there, you have more opening up that vesicle, more neurotransmitter comes out, then you're stimulating more and more and more. Another one could be blocking enzyme activity in the synapse. So now the enzyme, like let's say acetylcholine esterase, if we block that, then you're going to have a bunch of acetylcholine just chilling out in that synapse. And that's something that LSD, the drug named acid, would do. So it'll block the enzyme from breaking down the acetylcholine, and then it'll block the enzyme that breaks down acetylcholine. And then you get, I guess, hallucinations or whatever. And then it's pretty much the exact opposite on this side, but we do have one extra. First, we can block channels, right? If we block calcium channels, then calcium can't come in, it can't bind to the vesicle, and then the neurotransmitter can't come out. And so we block the calcium channel, then neurotransmitter cannot be released. Hopefully I've kept this video under 15 minutes. And then last here, we can actually block my friend over here, the sodium-potassium pump. So what does that do? That keeps the membrane potential. And if you remember from my graph of the action potential, well, it's not there. There it is. That graph there, if we keep that neuron non-polarized, whether we keep it at the hyperpolarized point, then it cannot fire another action potential. And that is a way to decrease the effect of the neurotransmitter because it can't release anymore. So the neuron cannot fire an action potential when it's hyperpolarized. All right, so that is the extent of the notes for the week. You will have a couple of worksheets based on this neurotransmitter and synapse stuff. I have some good ones about how drugs affect it. I have a interesting, I guess it's on the web. It's some fun thing to do. If you're using your iPad, you're not going to be able to do it, unfortunately. However, I took it, I did it, and I put it up on YouTube, and I have it in a playlist, so you guys will see that. I think that's on Wednesday, so that'd be tomorrow, I think. So we'll be using these. You should be using these notes, the neurotransmitter notes and the synapse notes coming up. Have a good day. I'll check in and say hello tomorrow. Peace.